Hello, my name is David Orren, and I'm going to talk to you for a few minutes about our UK Cares Innovation High Impact Board, the title of which was Exposure to E-Cigarette Vapor Alters Gene Expression and Induces Inflammatory Responses. Co-eyes on this project were Dr. Kevin Pearson, Dr. Holly Swanson, Dr. Chris Waters, Dr. Emma Demajwe, and Dr. Lee Chen. Our primary hypothesis for this study was that the vaporization of at least certain flavored electronic nicotine delivery systems, or ENDS liquids, basically vapes, would produce compounds that alter certain gene expression pathways, including those overseen by the AHR and NF-kappa B factors. And this would specifically happen in exposed cells in the lungs and the oral cavity, and there would be potential harmful effects, most notably increased oxidative stress and inflammation, which have been connected to activation of these two pathways. Our specific aim for to screen a variety of condensates created from ENDS liquids for their ability to activate these pathways and increase expressions of pro-inflammatory markers, increase oxidative stress, and increase DNA damage in cellular model systems. Again, these cells were mainly in the lung, from the lung and the oral cavity. In the second aim, we want to determine how heating and vaporization of ENDS liquids would be involved in promoting the harmful effect and to what degree that was. And lastly, we wanted to translate these studies to see whether we could see whether exposure of animals, in this case mice, to vapor from e-cigarettes would increase inflammation and injury in the lung, perhaps through these pathways, and connect these aims. So basically those were the three aims of the study. So after experiments examining these aims, we have just a variety of results I wanted to share with you. We first found that vapor condensates from at least several fruit-flavored e-cigarette liquids would generate cellular responses in our lung and oral cells that were consistent with activation of pathways governed by AHR and by NF-kappa B. And we know these pathways are, again, connected to oxidative stress and inflammation. Turns out it didn't matter whether the cigarette liquid, the e-cigarette liquid, contained nicotine or not, and the effects we've seen were independent of nicotine. We saw these effects in a reporter assay we have in our lab that measures activation of the AHR pathway. And with these fruit-flavored liquids, at least certain of them, we got more than a 10-fold induction in many cases, and this is substantial activation of this pathway, comparable in some cases to what we see with cigarette smoke. We got specific upregulation of AHR and NF-kappa B dependent genes when we looked at those in RT-PCR assays. We also saw transcription factor binding, in this work, the binding of AHR to its target promoter in gel electrophoresis studies. We also, using the COMET assay, were able to see increased levels of oxidative DNA damage in cells exposed to at least some of these fruit-flavored e-cigarette liquids. In regards to the second aim, we found that heating and vaporization was extremely important and actually necessary for the ability of these vapor condensates to activate the gene expression pathways we were looking at. Direct treatment of cells with the liquids that weren't vaporized was not effective in changing gene expression. You had to heat and vaporize these liquids to get these changes. And what that indicated was that sometime during the heating and vaporization, you're getting conversion to different chemical species in those vapors. That would mean it was those chemical species that was upregulating the pathways we were looking at. And so what that suggests to us, at least, is that the heating capacity of these systems that deliver the vapor, and there are many of those out there with different heating capacities, that's going to be an important variable in determining the toxic effects, i.e. the higher heat you generate, perhaps the higher toxic effect. Now, in our animal studies, well, that were done in Dr. Pearson's lab, we did repetitive exposure of mice to e-cigarette vapor, but that didn't significantly induce expression of the genes that were measured, and those are measured in lung tissue as well as kidney tissue, compared to mice that were not exposed. And we also had positive control in that which we exposed mice to cigarette smoke, which was indeed effective in inducing at least certain gene expression changes. And I should mention, with regards to these expression changes, we did see some changes in expression of genes in the vapor exposed mice, but they weren't significant, but they were in the same direction as the cigarette smoke mice, suggesting perhaps we need to modify our exposure regimens in future experiments. Now, there were some challenges associated with the project. A main challenge was COVID, which kind of disrupted the lab pretty much exactly at the time we were starting these studies, when the grant was awarded back in 2020. But on an experimental level, one of the key challenges that we faced, at least in our cellular studies, was generating condensates that were reproducible from one batch to another. And so it was really difficult to generate the e-liquid and the vapor condensates from the same e-liquid and have consistent biological effects from time to time. And we, presume that these were due to changes in the devices we were using over time. Perhaps they weren't generating the same level of heat. Perhaps our collection apparatus was not as good as it could have been, or our techniques were not as reproducible as it could have been. But our solution to this was, at least I think we think it's a solution going forward, at least, is now that the KTRDC, which is in the College of Agriculture, actually has a multi-channel instrument that's specifically designed for generating vapors and their condensates. And so we're certainly anticipating that when we get around to using this for generating condensates, we'll have a lot of consistent chemical composition, and that'll allow us to better compare our data between batches of the same condensate or between different condensates, and definitively determining the strength of the effects we're seeing when we treat cells with these condensates. Because of the animal experiments, as I mentioned, if we do more of those in the future, we're probably going to want to increase the animal numbers and or use longer or higher concentrations exposures to try to at least detect effects on animals that are being exposed to e-cigarette vapors. And I think with that, I'm going to just conclude the presentation and appreciate you guys' attention.